I'm going to let folks in. My name is Jess Delfiaco. I'm the communications manager for the Midwest Climate Adaptation Science Center. You are here for our monthly science seminar of May, and we are really excited to have three great presentations from affiliated graduate students at several of our consortium member institutions. Before we get there, I've got a few housekeeping notes and just a little background on the CASC for those of you who might be new or unfamiliar. We are recording this, so please keep your self muted and your video off if you would like during our students' presentations. We are going to have all three folks present and save our questions for the end of the hour. Please feel free to drop your questions in the chat throughout if you'd like, but know that you will have time to come on camera and share your question directly during the Q&A time at the end. So either way, share it in the chat or save it up and ask it directly. So for any of you who are not super familiar with the Midwest CASC, this is a map of our member institutions. We are a partnership between this consortium of institutions throughout the Midwest region, working in partnership with the USGS to produce science that can inform natural resource management efforts and help fish, wildlife, water, land, and people in our region adapt to a changing climate. And we are part of a national network of these regional science centers that all pursue that same mission. And we accomplish that mission in several different ways, including supporting a network of graduate students and postdoctoral fellows who are pursuing adaptation research, who you'll hear more from in just a few minutes. You can go to the next slide there, Sarah. A couple of quick announcements for our community. We've got a community of practice meeting coming up later this week, just a few days. For anyone interested, you do not need to be a formal member of the community of practice to join. This is for any climate researchers or practitioners who are interested. You're welcome to jump in. I think Sarah will pop a link to RSVP for that in the chat. And that's going to be focused on a conversation around data sovereignty and best practices in data use, led by Dr. Mike Dockery of the University of Minnesota here. Secondly, we've got our Summer Science Symposium coming up in just a couple of months, and we hope you all will join us there. That's going to be here in the Twin Cities at the University of Minnesota campus. And this is our opportunity for our dispersed community to come together in person, spend some time building connections, sharing science with each other, and just having some fun for a few days. So if you're interested in joining us, please check out our event page for more details and to register. Next slide, Sarah. Now, finally, we're going to go on to the stars of today's show. I'm super excited to introduce three students. Hannah Bay is going to be up first. She is a master's student working with Dr. Angela Burrow at Michigan State University. She studies the effects of shifting plant communities on the diversity and abundance of invertebrate communities in wet meadows and the impacts of those changes on the diets of native Enarins, frogs, yes, yeah, doing my best here. And she works to engage land managers and create adaptation strategies to address these changes in wetlands. And then we'll have Kaylin Wallen, who is a PhD student at the University of Illinois. She joined Eric Larson's lab in the fall of 2025 to work with the invasive rusty crayfish found in Midwestern lakes. And then Kesondra Key, who is a PhD candidate at Indiana University. She is going to share results from her recently submitted paper on a new way to detect plant water stress using data from 40 research sites across the United States. And that project explores whether changes in the timing of plant water use can provide an early signal of drought stress. So with that, I'll turn it over to Hannah to get us started.
Perfect. Thanks so much. Let me share my screen. Is it presenting?
Yep, it looks good.
Perfect. I'm going to move this. Okay. Hi, everyone. Today I'll be talking about the impacts of changing plant communities on invertebrates and Enarins in wet meadows. So to start off, we know that plant communities are shifting due to a variety of stressors. Some of these are natural, such as natural disturbances that lead to succession, but a lot of them are human-mediated nowadays. So those can be land use changes, invasive species, and global climate change. And for global climate change specifically, some changes we can see are changes from specialist species to generalists within communities. So we're losing some of those specialists, as well as changes in the functional trait structure and diversity of plants. So those are characteristics related to the plants. And we're seeing more plant communities related to things that are like highly drought tolerant or better at getting resources quickly. So we're seeing overall changes in the structure and diversity of those traits as well. And a lot of conservation goals are focused on increasing resilience to these stressors so that not only the plant communities can survive in the face of these stressors, but also because changes in the plant community have direct impacts on other groups. So this is a bit of a busy figure from Scholl et al., but notice how changes in the plant species richness and functional diversity, as well as the traits of those plant communities, directly impact herbivores and other taxa within the group. So we know that as these plant communities are changing, it's also affecting other groups. This is really important in highly stressed communities. An example of that are wetlands. Wetlands face a lot of degradation worldwide due to all of those different stressors I mentioned. And so changes in their structure and plant communities are expected to change the invertebrate communities present. And while bugs are great, and that's lovely, we care about other groups as well, such as anurans or frogs, which use these bugs or invertebrates as their food resource. But unfortunately, we don't know much about how changes in those plant communities are actually affecting their diets as a whole. So my research focuses on how changes in these wet meadow plant communities affect the plant community characteristics in themselves, and how those changes in characteristics affect the food for anurans in the community, as well as the diets that we actually see for the anurans. So my presentation will go through each of these three questions as a whole. Starting with the plant communities, I looked at 8 different wet meadow sites in mid-Michigan. So this is my little map with the stars each representing a pair of sites. And in each pair, we had one high-quality site and one low-quality site. The way we determined quality was through the Michigan Natural Features Inventory use of what they call an eco-score, in which high-quality sites had high eco-scores, meaning they were not very degraded and not very invaded, where low-quality sites were very invaded and very degraded overall. In these 8 sites, I looked at the plant communities and characterized the percent cover of each species present. And from the dominant species, I collected leaf clips to look at their functional traits. And I'll talk a little bit more about which functional traits we looked at a little later. From the different plant communities, we wanted to look at their characteristics. So other than just what species were present, we also looked at quality metrics. For those who are not botanists, C-values, which are used both in the weighted C-value and floristic quality index, are a number given to a plant describing how high quality habitat it needs to survive. So a 10 would mean it's a very specialist plant, needs really high quality habitat. And a 0 or a low number would mean it can live anywhere or it's invasive. So we looked at these quality metrics in each of the plant communities. We also looked at diversity metrics, so how many species were present and what was the proportion of those different species to one another in the community, as well as compositional metrics like how many invasive species were in the community and what was their cover. So we looked at all of these different characteristics in an NMDS graph. So if you aren't familiar with NMDS, it takes this multidimensional data and puts it into 2D. So in this graph, each of the dots, so these 4 red dots and these 4 blue dots represent my plant communities or my sites. And each of them are taking the information about the proportion of different plants in those sites and putting it into a 2D graph. And I also put some of those important characteristics we discussed on the previous slide in there as well. So we can see in my low quality sites, they have a higher percentage invasive cover and a lower weighted C-value or lower quality overall. We can also see that the lower quality sites are taking up less of a trait space, meaning they're more homogeneous or more similar to another than the high quality sites, which are more heterogeneous across the landscape. So now we know that there are a few differences in the plant communities, but we want to know what that actually might be doing for the insects or the invertebrates. To survey invertebrates, I use pitfall traps. This is a example of one of my sites. I put the pitfalls around the perimeter of the site. I was only able to do this at 7 of my 8 sites since one was too wet to set up pitfalls. And I kept them open for a week, which was at the same time that I was surveying the frogs in the site. From each of these pitfalls, I looked at the number and abundance and diversity of the invertebrates in the community. And I wanted to see how the different characteristics of the plant community we discussed earlier affected those invertebrates. So those are the other characteristics we talked about, but I also wanted to know how those different functional traits, which I mentioned earlier, also affected the community. So these functional traits we looked at are down in the blue box. So that was the C to N ratio, which tells us how high quality of food the plant could be for an invertebrate. So a lower C to N means it's a higher quality food. The height or structure of the community tells us how much living space they have, essentially. Like a higher community might have more plant mass for them to live in, biomass. And total phenolic content is a measure of plant chemical defenses. So when there's higher phenolics, it means it's higher chemical defenses, which is usually worse for invertebrates. So looking at all of those different plant community characteristics and community-weighted mean functional traits, we wanted to see how they, in combination, had effects on the invertebrate community. Unfortunately, a lot of these different characteristics are extremely related to one another, and we had high levels of collinearity. So I couldn't do simple linear regressions. Instead, I had to take these characteristics and put them into new variables through PCA. So this is part of my PCA, or principal component analysis. In the graph, we have our 4 low-quality sites and our 3 high-quality sites, and I put those same characteristics that we put on the NMDS in here, except now it's showing us how each of these different axes combine characteristics that are collinear to one another to make a new variable that we can use in linear regression. So our first axis explains 58.8% of the variation of the plant community, and it is described by the invasive cover, the C to N, the weighted C value, and the FQI. So basically, the first axis is explaining the plant community quality and the plant community composition as a whole. Our second axis looks at the plant community diversity through the species richness and the diversity, as well as the community structure through the height. And a third axis, which we can't really see here, but that does exist, is explaining 9.7% of the variation, which is the chemical composition or the total phenolic content. So now that we have all of these variables into 3 main variables, we can use them to explain linear regression with the responses of the invertebrate community. So we looked at these different components on the invertebrate abundance, but did not find any real relationships between the plant community and the abundance of invertebrates. However, there were some implications for diversity. So we saw that PC2 showed up to be important, which was showing that as the plant community diversity, richness, and height increased, we also saw increases in invertebrate diversity. There's also a trend with increasing total phenolic content correlating to increases in invertebrate diversity as well. So we're seeing that the changes in the plant community, or like the structure of the plant community, is having some effect, at least on the diversity of invertebrates. Now we wanted to see if those changes were also reflecting in the diets of the frogs. So to capture frogs, I did this at the same time I was capturing invertebrates. I did nighttime surveys, captured frogs, and used a process called gastric lavage to collect their stomach contents. It's basically stomach flushing. The frog lives, they're not happy about it, but they'll be okay at the end of it. We collected 39 frogs from high-quality sites and 30 from low-quality sites. My first question is, were they actually eating what was available to them in the community? So I used Kendall's tau, which looks at the correlation between the availability of food in the environment. So my little picture over here shows an environment where we have 3 orange insects, 2 blue insects, and 1 yellow insect. And we can see that in this case, the frog is eating those insects or invertebrates at the exact same proportion as what's available in the environment, which means that the Kendall's tau would be significant. And that's what I saw in my cases. So in both the high quality and low quality sites, they were eating what was available to them in the environment, which is super important because that means if we can see changes in the invertebrate community, we expect that to change the diets of the frogs. I also looked up what was making up the diets of each of the frogs, or the frogs in the high and low quality sites. In the high quality sites, they were eating mainly coleopterans or beetles, followed by invertebrate larvae, then worms, then gastropods, and flies. In low quality sites, we saw similar groups, but a little bit different orders. So they were eating mostly gastropods, followed by beetles. We had isopods pop up in this as an important food factor, as well as harvestmen and grasshoppers. I'd also like to point out in the high quality sites, they had slightly higher percent occurrences overall. So they seem to have slightly more similar diets in the high quality sites compared to the low quality sites. They were just eating a little bit of the more of the same thing. So putting that all together, we saw that there are shifts in plant communities and that these shifts can impact the community characteristics as well as the functional traits of the plant communities. We're also seeing that those shifts might be influencing the diversity of invertebrates but possibly not their abundances in the site. We also saw that changes in invertebrate communities are likely to influence the diets of endurance since they're eating at what is proportional to them, what is proportional in the environment. Moving forward, some management implications for my research. First, there's value to all sites for restoration. Even my sites that were highly invaded or really covered in reed canary grass had a good proportion or a good number of important invertebrate community in taxa. So it's important to focus on these sites as well for restoration and not throw them out. Another important thing is to favor management goals that increase native diversity in the plant community. We saw that the diversity of the plant community, rather than their overall quality or number of specialists, seemed to be more important. So using diverse seed mixes of adaptable and generalist natives are good options, both to help the invertebrate community and also to prepare for the adaptation to climate change. And it's also important to support microhabitats in wet meadows. Carex stricta is a sedge that's super important to wet meadows, and it's super cool because it makes these little tussocks, which you can see in the picture, and it allows other plants to live on them in other pretty wet communities. So it increases the diversity locally of a lot of species. So in focusing on the restoration of this specific species can help the overall restoration of wet meadows and their diversity overall. So next steps, I'd like to look at differences in the invertebrate community compositions within the communities. So not only what is changing about the diversity, but like what groups are actually popping up to be more important in the high quality versus low quality sites. I'd also be interested in learning more about other plant community traits that might impact the invertebrate communities outside of the 3 functional traits I looked at, especially those traits we really expect to change with stressors like climate change. I have a lot of people to acknowledge. First, my advisor, Dr. Burrow, as well as technicians and undergrads in our lab who helped with this research, as well as the CASC. It's been great to be a part of this community, both for their support financially, but also a lot through the community, through the mentorship program and other opportunities to learn a lot as a growing scholar. So I appreciate the CASC and everything they do. And thank you so much.
Thank you so much, Hannah, for that great presentation. Reminder for everyone that we are going to let everyone present, our 3 presenters go, and then we welcome questions. If you have a question, a burning question for Hannah that you don't want to forget, please feel free to pop it in the chat and we will come back to it at the end. But I am going to invite our next speaker up. Kesondra, are you ready to go?
Yes, I can go next.
Fantastic.
Sorry, let me just share my screen. Sorry, one moment.
No problem.
Okay. Here we go. Sorry, I had to fix my headphones there for a second. Okay.
No problem. It's always the hardest part of any Zoom is getting the
Yeah.
finger slides up on the screen. That looks great.
Okay, looks good.
Yep.
Okay. Let's see. Okay, so hi, my name is Kesondra Key. I'm a PhD candidate in the School of Public and Environmental Affairs here in Bloomington, Indiana. And I am, of course, an MCAS fellow. Today, I'll be talking to you about our new plant-based drought signal, where our paper on this topic is currently in review. During this talk, we will briefly go over why plants are important, how drought unfolds, and then pivot into our plant-based drought signal with a little on our methodology, results, and end with our next steps and some take-home messages. To start off, plants are not simply static pieces of the landscape. Plants are active participants within the Earth system, providing carbon storage through holding carbon within tissues like tree trunks, branches, leaves, and roots. Water regulation by moving water from the soil back to the atmosphere through transpiration, protecting the soil from erosion, cooling the planet through water vapor via transpiration, biodiversity support through habitat creation and support, and of course, providing food, fiber, and medicine. But how can we look to plants to understand where drought is occurring and how ecosystems are responding? Some existing drought indicators already focus on plants, where some rely on changes in vegetation greenness, like NDVI, which can show when leaves are browning or wilting. These indicators are useful, but these changes may appear after plants have already been quote unquote stressed for some time. Before we get more into this, let's first walk through some key steps as the drought unfolds. So for the next few slides, let's walk through a simplified sequence of how drought progresses through an ecosystem. First, drought begins with a rainfall deficit, meaning precipitation is lower than normal. At the same time, atmospheric demand can increase. Warmer and drier air increases vapor pressure deficit, or VPD, which is a measure of atmospheric aridity. This is going to come up again a bit later. As low rainfall continues and water is still being lost through evaporation and plant water use, soil moisture begins to decline. These meteorological and soil moisture changes can happen over hours to days. With lower soil moisture and often higher atmospheric demand, plants become constrained in their water use. One way they can respond is by regulating their stomata, which are small openings on the leaves that can control water loss and carbon uptake during photosynthesis. Under more stressful conditions, plants may shift when transpiration peaks, with the peak occurring earlier in the day rather than near midday when solar radiation is at its highest. I'll explain this timing shift in more detail a little bit later. As water becomes more limited, plants reduce how much photosynthesis is happening, which can also limit carbon uptake and thus lower productivity overall. As drought continues, ecosystem and societal impacts can become more visible. Plants may show browning, yellowing, reduced growth, and even wilting. There may be impacts on agriculture, water resources, and overall ecosystem function. By this point, the timescale has shifted towards weeks to months. Many traditional drought indicators, such as SPEI or PDSI, are essential for monitoring drought, but they often reflect drought after these broader physical or visible impacts have developed. The key opportunity is stage 4. Plants may adjust their water use before visible discoloration or productivity declines are obvious. So instead of focusing on hydrological or meteorological variables like precipitation and soil moisture, or ecosystem or societal impacts like lower water availabilities in reservoirs, we have an opportunity to focus in on if we can detect quote-unquote plant stress at this stage. As a quick recap, traditional drought monitoring often focuses on meteorological and hydrological variables. My goal is to add a biophysical perspective by using plant timing signals, specifically the timing of evapotranspiration, which is both transpiration and evaporation of water out of an ecosystem, to detect emerging plant stress during drought, which we will look at in the next slide. Okay, now let's shift to the basis of our plant-based drought signal. In stage 4, I mentioned that plants can respond to drought through stomatal regulation, and that this can shift the timing of transpiration and thus evapotranspiration. This simplified plot shows ET over the course of a typical day. The y-axis is relative ET, and the x-axis is the hour of the day. Under wet conditions, when water is less limiting, ET tends to increase through the morning and peak around midday when solar radiation is at its highest. Under dry conditions, however, this peak can shift to earlier in the day. As water becomes more limited and afternoon conditions become hotter and drier, plants may regulate their stomata to reduce water loss during the most stressful part of the day, which can result in an earlier peak ET. So the key idea here is that the timing of peak ET may contain information about quote-unquote plant stress responses to lower soil water availability. To quantify this earlier peak ET timing, we created the diurnal decoupling factor, or the DDF. Even though the name sounds technical, the calculation is simple. It compares the timing of peak ET with the timing of peak VPD, where we use VPD as a reference point since it is a purely atmospheric variable. ET usually peaks before VPD, which typically occurs around 4 p.m. regardless of drought conditions. So the key question is not whether ET is earlier, but how much earlier it peaks relative to atmospheric demand. In this example, from an Arizona flux tower site, relative ET is shown in blue and relative VPD is shown in orange. This plot is depicting a real drought in 2013, which shows earlier ET timing while VPD peaks later in the afternoon. This timing separation is what the DDF captures. In short, the idea here is that a larger DDF is indicating quote-unquote plant stress or that earlier peak ET timing. This brings us to 2 of our main research questions for the study, which are as follows. Does the DDF shift towards more negative values or that earlier peak ET during soil moisture deficit? And does the magnitude and direction of the DDF shift vary with drought severity? To answer these questions, we tested the DDF during drought versus non-drought conditions based on low observed soil moisture conditions and by drought severity across 40 eddy covariance sites within the United States, along with grouping these based on vegetation type and by humid versus arid sites. On the right here, you can see that the Eddy Covariance Tower that's located near Bloomington, Indiana, within the Morgan-Monroe State Forest. This is just an example of what these towers look like. This one is managed by my advisor, Kim Novick, here in Bloomington. If you're not familiar, Eddy Covariance Towers use specialized sensors to measure exchanges of carbon dioxide, water vapor, and energy between ecosystems and the atmosphere, along with environmental variables like temperature, relative humidity, soil moisture, and many others. This plot shows the main pooled result across all 40 sites. The x-axis is daily DDF in hours, where more negative values indicate that ET is peaking earlier relative to VPD. The red box plot represents drought days, while the blue indicates non-drought days. During drought, the distribution shifts slightly to the left, meaning that the DDF became more negative. Overall, ET peaked about 30 minutes earlier on average during drought, with the caveat that our eddy covariance data is half-hourly. This is a small shift, but it was consistent across the pooled dataset and statistically significant with a Welch's t-test p-value below 0.001. In short, our pooled results support that the DDF is a plant-based drought signal with a small but significant signal. Next, I categorize drought based on severity. Here, blue represents non-drought, yellow represents less severe drought, and red represents more severe drought. Again, more negative DDF values mean ET is peaking earlier relative to VPD. The letters on the right show the two key post hoc groups, so groups with different letters are statistically different from one another. In this case, all three groups are significantly different. The less severe drought group shows the strongest earlier timing shift, with the distribution shifted furthest to the left. This suggests that earlier ET timing emerges under drought stress. However, the more severe drought group moves back closer to non-drought conditions. This was surprising because we might expect more severe drought to create an even stronger shift. Instead, the response here appears to weaken under more severe drought. So the key here is that the DDF does respond based on drought severity, but it is not linear. So why does the more severe DDF look more like non-drought conditions? To think through this next part, remember that ET includes both transpiration from plants and evaporation from soil or surface water. Under more wet conditions, transpiration can make up a large share of evapotranspiration, especially during the growing season. So the timing of ET is more strongly influenced by plant water use. But as drought becomes more severe, where water becomes even more limiting, photosynthesis may decline, reducing how much transpiration is actually occurring. When this happens, evaporation may make up a larger relative contribution to total ET, so more transpiration than evaporation. More evaporation than transpiration. Alternatively, transpiration may be so limited that the DDF no longer captures that shift clearly. It is also possible that the overall magnitude of ET is simply too small to effectively detect these changes. Therefore, the key takeaway is that more severe dry conditions can weaken the plant timing signal that the DDF is designed to detect, reducing the DDF sensitivity to plant stress under severe drought. To summarize the results, the DDF showed a small but consistent earlier ET shift during drought. Across the pooled dataset, ET peaked about 30 minutes earlier during drought, with the caveat that these are half-hourly eddy covariance data. While we didn't have enough time to go over all of our results, this pattern was observed across vegetation types and aridity classes, with the strongest shift occurring under less severe drought, while weakening under more severe drought. So overall, the DDF appears to capture a consistent plant water use timing response during drought, although the strength of that response varies with drought severity. Because the DDF relies on eddy covariance tower data, one challenge is that it is spatially limited. So the next step is to ask whether similar ET timing signals can be detected remotely using satellite. For our next steps of this project, we plan to utilize land surface temperature to upscale the DDF. Since transpiration cools the land surface and our satellite data provides two daily timestamps, one earlier in the day and one closer to the afternoon, we can look at the differences in LST during drought versus non-drought and to see if the signal works. The two maps here are showing only vegetated land cover and are a rough proof of concept from the 2012 drought at the Morgan-Monroe Flux Tower site, which is depicted by the yellow diamond. The map colors indicate LST, where blue is cooler and red is warmer. Here we see LST on June 7th, 2012, where we see cooler temperatures in the afternoon, while by June 15th, when the 2012 drought was well underway, The pattern appears to shift to cooler temperatures in the morning, which could be consistent with an earlier ET timing captured by the DDF. It's important to note that these maps are two hand-picked examples for what our plan is moving forward, so that this is not a proof of concept just yet, but it showcases that there may be promise in utilizing LST for the DDF upscaling approach. So why is this important, and how could we use the DDF to support climate adaptation? As we saw in the 2012 drought example, drought impacts can be devastating, with billions of dollars in damages across the United States alone. One challenge is that many traditional drought indicators can take weeks to months to fully signal that drought is developing, often after it is well underway. The DDF offers a different perspective because it focuses on plant timing responses. Instead of only looking at atmospheric dryness, soil moisture decline, or visible losses in greenness, the DDF asks whether plants are changing the timing of ecosystem water loss as stress develops. In the study, we're not directly detecting whether the DDF can predict drought onset earlier than other signals. Instead, we're looking at how the signal behaves during drought overall. But the potential application is that the DDF could eventually contribute to earlier warning frameworks or provide additional information within existing drought monitoring systems by indicating that plants are already responding to moisture stress. We showed that this signal can be monitored at point locations like eddy covariance tower sites with potential to upscale using satellite observations, which is our next step. More broadly, the DDF has a potential to complement drought detection by including temporal biophysical responses of ecosystems, not just the physical draining of the environment. For landowners and natural resource managers, earlier information about plant stress could support earlier decisions, such as prioritizing mitigation efforts or allocating water resources more carefully. So while this management potential is beyond the scope of this paper, it highlights an important opportunity for where the DDF could go next. The main take-home messages is that plants can reveal drought responses through shifts in ET timing. In this work, the DDF captured a small but consistent shift towards earlier ET timing during drought, with the strongest response under less severe drought. Finally, while this work is flux tower based, future work can test whether this similar timing signals can be scaled using satellite land surface temperature. And overall, plant timing signals provide a biophysical perspective on ecosystem drought response. This work is currently in review in JGR Biogeosciences, and we're looking forward to reviewer feedback soon. And that brings me to the end. I want to acknowledge my advisors, Mallory Barnes and Kim Novick, along with our funding sources, since I've been a PhD candidate, which is NASA, NSF, and right now I'm the Midwest Climate Adaptation Center, which I very much would like to thank and appreciate. So thank you for listening, and feel free to contact me via email or for questions after the seminar.
Great. Thank you so much, Cassandra, for a great presentation. I am checking our time here. We're doing pretty good, but I want to swiftly move us along to Kaylin. Kaylin, are you ready to take over the screen? And then just as a reminder, folks, you're welcome to drop questions in the chat or save them for our Q&A after Kaylin's presentation.
Yes, I am. Let me share the entire screen. Alrighty, let's start at the beginning. Can you all see?
Looks good.
Alrighty. So, hello everyone. Today I'll be giving you brief insight into a review I'm doing on the effects of warming winters on freshwater non-native invertebrates in North Temperate lakes. So first, we have to understand, which many of you all already know, that climate change is impacting lakes worldwide. It's causing rises in temperatures, altered watershed hydrology, declining lake levels, and harmful algal blooms. And this is of particular concern for North Temperate lakes in the Midwest, where climate change research has focused on the open water season or the summer season instead of winter, despite the carryover effects winter has on these lakes in the summer. And this is particularly concerning because North Temperate lakes experience strong seasonality, and reduced winter severity is a key effect of climate change on North Temperate lakes. And in the Midwest, this is a major concern once again because we rely on the lakes for jobs, income. It's a water supply for many of the major cities and for agriculture, fisheries, and things like that. Warming winters may also affect the establishment and spread, impact, and management feasibility for many of these non-native invertebrates, and we'll get into how this may affect them later. So the objectives of this review, or some questions that we have, is how are non-native freshwater invertebrates affected by winters? We want to know what biological processes are being studied most on their winter ecology. And of this, we want to know which of these freshwater invertebrates are best studied. So the effects of warmer winters on lakes is something I specifically had to look into, and I focused on three main areas. So the thermal stratification timing is changing. There's later fall mixing and earlier spring stratification. The timing of ice, or the ice phenology timing, is changing. Precipitation patterns during the winter is altering or changing throughout the season, and this is causing lower ice quality and later ice formation in the fall or early winter, and then earlier ice formation or earlier ice off in the spring. And there's also being a timing change in the dissolved oxygen dynamics. There is to be assumed a shorter stints of hypoxic events during the winter and longer summer hypoxia or summer kill. So I'm doing a bit of a systematic review. So to do this, I needed to create a species list, and I did this using the USGS Non-Indigenous Aquatic Species database, where we took 4 invertebrate phylotaxa, and for our inclusion criteria, we really wanted to make sure that we are getting established invertebrates or non-native invertebrates within the Midwest states or Midwest CASC states. And of that, we started with 231 species. I also wanted to include possible high priority species from the Lacey Act, which left us off with about 231 in total. And for our inclusion criteria, not only had it to be established within the CASC states, we also wanted to make sure that they were freshwater species and had an active taxonomic profile at the time. And we included only 4 of the landlocked Great Lakes, which resulted in a total of 79 species. For rendering papers, we're doing a literature search back into the 1900s. So I wanted to account for all the different name changes, scientific name changes in the past. And for this, we did a taxonomic profile on the World Registry of Marine Species to include these names in a search string, which we have, for example, at the bottom. And we included the terms lakes and winters to try to get the broadest search possible for each of the species. So as a result, we got 127 papers, and each paper's title, abstract, and full text was reviewed to determine relevancy. And for relevancy, we wanted the study to either be conducted during the winter, contain relevant winter ecology on the target species, and focus on lakes. We did not include any other bodies of water like rivers, river basins, streams, and wetlands. And I have 2 examples of excluded papers on the side here. We have T. crassus that was excluded because the study, although it focuses on the target species, it was conducted during the summer. And then we have another paper that was picked up, although it did mention our target species, it only mentioned it in the references. And 2 examples of papers that were included was a paper on our zebra mussels, in which their reproduction was surveyed throughout the seasons, and then a study on Corbiculifolia that was a lab study that looked at how temperature regulated their excretion rates. For our results, our zebra mussels had or returned the most papers. So out of about 90 included papers, zebra mussels returned 43. And most of our other species only returned less than 5 papers, with many of them returning only 1 or 2. And out of 79 species, only 22 of our non-native invertebrate freshwater species were represented. meaning that 2/3 of the species had no relevant winter ecology, despite the fact that this included 3 mitten crab species that are potential high risk, and then other high impact invasive freshwater species like our rusty crayfish and our Chinese mystery snails. For being that this is a systematic review, I really wanted to categorize the papers further, and I did these by the lake processes that I further mentioned before in our biological focus areas. Temperature was featured throughout the studies, and some of the papers I included it separately because temperature, some of the papers only focused on temperature and not any of the other lake processes. And then life history was studied the most biologically throughout all of the papers as well. For a brief summary, of course, our zebra mussels were most studied, and it really shows that a lot more winter limnology studies need to be done across the invertebrate taxa, and temperature, and many of the studies link temperature to different life timing or life cycle processes, showing that warming winters will definitely cause shifts in a lot of the life cycle timing of these species. And out of the species, not all of the freshwater species reacted the same to warming winters. Some were more advantaged or disadvantaged than others. So as winters warm, our kind of invertebrate taxa or non-native invertebrate taxa will be have different effects or be impacted differently by warming winters. A pretty neat example of this throughout the literature so far is that our quagga mussels may outcompete our zebra mussels as these conditions continue to change throughout as winters warm. And this is winter limnology is a key change from climate change, and it's really important because we need to know how to manage and possibly predict some of our future invasive or non-native range shifts as time goes on. Well, kind of going back to this briefly, I am also currently working on synthesizing some of the changes based on papers or based on species that had more than three papers to kind of see how warming winters will affect them. But this is taking a bit more time, and I'm working on some figures throughout their life processes as well. So for acknowledgements, I really want to thank the Midwest Climate Adaptation Center. This would not have happened without them, including a lot of mentorship opportunities through them. I was able to get some pretty good feedback on the research and things that I can further look into and bring into my rusty crayfish research. And of course, thank you to my advisor and all of these other lovely people that helped me do this systematic review.
All right. Thank you so much, Kaylin. If all of our speakers want to come on camera, we're going to open up the Q&A. I'm going to hand it over to Dan to facilitate that.
Thanks, Jess. Thank you all. Those were wonderful presentations, really interesting and wide-ranging stuff. And yeah, for questions, anybody is welcome to come off mute, turn on their camera, and ask. While we wait for people to present their questions, we did get one in the chat for Hannah already. And the question says, while fully recognizing the difficulty in identifying partially digested invertebrates, do you think you'd start to see differences in availability versus selection if you were able to identify them beyond order, down to genus or species? And if doing so, might there be more identifiable differences between low and high quality sites?
Yeah, thanks. That's a good question. You're right. It's really hard to get below order or sometimes family level within digested invertebrates. I could see it going either way. I do know one of our bigger categories was just invertebrate larvae as a whole, so like grub-like creatures or grub-like invertebrates. I do feel like, at least from what I was seeing characteristically, the difference between which types of invertebrates we were catching in the pitfalls, at least in the pitfalls, compared to the diets, might differ a little bit in terms of larvae. There seem to be more Lepidopteran larvae within the diets of anurans than what I remember catching in the pitfalls specifically. That being said, when you go to family level for like beetles, we caught a lot of crabidie, so like the ground dwelling beetles, and I saw a lot of consistency in those beetles at least, which seems to be a really important food source between the pitfalls and the diets. Going down to species, I'm sure we'd start to see some selectivity, but things at family level might stay a little bit similar. So, yeah, I think at family maybe the same, but then as you go to species, it definitely we could see some selectivity occurring.
Great, thank you. Kaylin, I had a question for you. And I'm not sure if this is something you kept track of during your review, but if you have a sense for it, the studies that did have the types of information you needed, were they studies that were motivated by understanding impacts of climate change, or did they tend to be other types of ecological studies where they kind of happen to have information you could use?
So many of the studies were done in the early 2000s. So when those climate questions were just being like formulated into research. So I'd say a good half of them were trying to relate warming winters to these invertebrates. But of that, I'll say half of them were on zebra mussels. So that really was majority of it, looking at how climate is going to affect our zebra mussels, I'd say most of which. And the others were really just looking at those temperature related life cycle dynamics that were shifting or changing, or predator relationships from waterfowl predating on some of these species and things like that.
Great, thank you. And I'll stick with you, Kaylin. We got a question for you in the chat from Olivia. What was the most challenging stage or part of your review process, or were there things that surprised you and were unexpected?
I think the most challenging stage was categorizing the papers between their like focus areas or biological focus areas. Being that like it's some papers really debated on whether this was life history or was this really a predation or a reproductive process and things like that. So categorizing the papers and having to go over and read some of them and recategorizing when some like, no, I found a new category to put this in. I think that was the most intensive process, is spending winter break reading the papers and doing that part. But a surprising aspect of this was learning that these invasive species were really going to be impacted and their range will be changing. And I was not expecting that there were to be interspecies changes within these communities. And I think that was the most surprising aspect, is not only is it affecting that one species, but the species interaction. So I thought that was really cool.
Great, thank you. Kesondra, I have a question for you, and I know you said that there's a lot of stages, many steps to go before using the DDF as a drought predictor. But if sort of all went well with the proof of concept and developing this, how much quicker of an early warning system for drought could this potentially provide?
That's a really good question. I'm not sure I have an exact answer, since there's more work to be done to see how much sooner that actually will show. But as a point here, the DDF is a subdiurnal signal, so we can look at this per day as opposed to many of the other variables, which can take weeks to months to really start to see that drought is happening. So I think there is some pretty good opportunity here for that. But yes, there is more research that needs to be done on it. I see there is a question.
Yeah, and another question for you, Kesondra, from the chat from Chris. Were you able to look at how DDF varied across tree species, noting that tree species vary somewhat in their stomata responsiveness? And Chris was wondering if that was detectable.
Not super specifically. We did look at deciduous broadleaf forests versus evergreen needleleaf forests. I think they both had, I can't remember specifically, but I think they were both pretty similar, with like the DDF going earlier during less severe drought as opposed to later, but we didn't look any closer than that for vegetation type. But we did think about it though. I hope that answers your
Now
question.
I'll leave this space open for anybody who would like to unmute and ask their question. Got a shy group today.
I think it's because it's Monday.
Yeah.
Okay, I've got one question. I'll jump in here. It's a silly question, but I wanted to ask. Hannah mentioned that the frogs survived but didn't like the gastric flush. I was wondering how she knew they didn't like the gastric flush. I mean, maybe they're weird, maybe they enjoy it.
Great question, Chris. Thank you. I could tell they didn't like it because a lot of the big ones physically remove the tube, and it was actually a real challenge. You have to like pin their arms down, because they're pretty strong, which is surprising. So I could tell they didn't like it because they were trying to get out of it.
Nice.
Jessica.
Hi, everybody. I was kind of holding on to my question because it's not really a science question. I'm wondering, each of you, it's more of a CASC in your research in relationship to CASC question. Each of you acknowledged the CASC and suggested that it was somehow valuable to you financially, yes, but maybe in other ways. I'm wondering if you'd either elaborate on that or say how you think the CASC could be helpful to you now, like in the next stage of your work, or what it might mean to you professionally. What is it doing for you, or what could it do for you since you are associated with this organization? It's kind of an open-ended question.
I can go first. I kind of mentioned this during my presentation a little bit, but I think one of the really great things the CASC has done, at least this year, is I participated in the mentoring program actually with Dr. Hoving, and I feel like I learned a lot through that, just the ability to do some professional networking and meet with people who are actually in the field doing jobs that are important to us is really important. I think also the CASC is just a wonderful opportunity to meet people with a general interest in climate adaptation, but who are doing a variety of different things. I mean, all of our talks were very different today, and I think that's great. It allows for collaborations that you wouldn't otherwise expect, because otherwise, like Angela and I are at herp conferences or something, just talking about frogs, and that's great. But it's also great to see people who are broadly interested in these larger issues, and I think that dynamic is really important as well for going forward in collaborations for future research.
I can go next. I 100% agree with that. The mentoring program I also was in with Dr. Robertson Thompson, and that was very illuminating. She's from the USGS, and just, I don't know, being able to talk to someone in the industry and like how they do things and how they got there. And like, I don't know, it's just been really fantastic. The conference last year was very helpful. I had a poster presentation and got to talk to a lot of different people. Same kind of thing, like conferences I usually go to, like might not have as a wide variety. The workshops have been really helpful. There was one about presentations, I think, recently, which had some really good tips. Moving forward, I mean, this kind of thing was very helpful to get me out of my comfort zone, practice, and do these presentations, get feedback is excellent for early career. I don't know, just job opportunities, talking to people, seeing where climate adaptation opportunities are, like all of it's been pretty fantastic. That's it for me for now.
So I definitely say that I really enjoyed the coffee hours. I think it was very helpful to get kind of modern research outlook, different conversations and discussions were happening within the field without just analyzing some papers or some research talks on my own. I really had I was able to have a conversation and get other opinions on some of the work, and some of it really helped with like research and just my classes and things like that. I think as a first year PhD student, I think these different mentorship and the workshops really helped. It got my feet wet and got me introduced to kind of thinking like a scientist outside of my bachelor's. And I also really shout out to Dr. Alex Hotska, my mentor. He really gave me some insight on my research and different kind of like early career things to think about when I'm doing some of these projects, and got insight on some of my summer research, and just it was very helpful.
Great. Thank you all for that feedback. That's really helpful.
Yep.
All right. Well, thank you, Kaylin, Cassandra, and Hannah once again for your excellent presentations. And I'm going to turn things over to, and thank you all for coming today for this seminar. And I'm going to, in closing, turn things over to Jess. Close us out.
Yeah, ditto. Thank you, Dan, for the Q&A. Thank you to these great presentations and for everyone for joining us. Quick poll before you hop off, super helpful for us. And then as you can see on the screen here, we'll be back a month from now, the fourth Monday in June, for another seminar. We hope to see you there. Thanks, everybody. Have a great rest of your Monday.